Okay, so now that we talked about the supporting cells within nervous tissue, depending on whether or not it's the central nervous system or the peripheral nervous system, let's talk a little bit about neurons themselves. So these are the structural units of the nervous system. They're fairly large, they're definitely specialized so that they can conduct impulses, meaning that they're excitable. They have several special characteristics. So one is long extreme longevity. So most of them can last a person's lifetime as long as you remain in good health. They're amitotic, meaning that they lose their ability to divide, and we cannot replace them if they're severely damaged. There are a couple of exceptions. So we do have some neurons in our olfactory, so in our nose area, and our hippocampus. That can undergo division, but the majority of them are amitotic and can no longer divide. They also have a high metabolic rate, so they require a continuous supply of oxygen and glucose. They cannot survive long without oxygen at all, and that's because we're constantly moving ions across the plasma membrane, which means if we are moving ions, most of the time it's going to require some sort of energy, and in order to produce the energy in the form of ATP, we have to undergo cellular respiration using the oxygen and the glucose. We talked a little bit about that when we talked about skeletal muscle and production of ATP. When we talk about the structure of a neuron, they all have a cell body, that's essential, and then they have one or more processes. So the cell body of a neuron we can also call a soma, and this is the biosynthetic center of a neuron, um, and also the metabolic center. So we have a high number of mitochondria in the cell body. What's its function? Basically, going to synthesize proteins, membranes, and chemicals. We have a rough ER, which contains what we call Nisl bodies. Those are the ribosomes that actually stud the endoplasmic reticulum themselves. You'll have to identify those structures in lab. They also contain spherical nucleus, and they still have their nucleolus. There's also some pigments such as lipofuscin and black melanin, and in most the plasma membrane is part of the receptive region, meaning that it's going to receive input from other neurons. So if we're talking about cell bodies of a neuron, depending on where the cell body is located, determines kind of what we call it. So they typically are found in clusters. So if we have clusters of neuron cell bodies in the peripheral nervous system, we call those nuclei. If we have clusters of these cell bodies outside of the brain or spinal cord, so in our peripheral nervous system, we refer to them as ganglia. So when we hit chapter 12, you'll have to learn a couple names of specific ganglia. And again, that's just referring to collection of the cell body of a neuron in the peripheral nervous system. So we said that every neuron has to have one cell body or soma, and then it can have one or more processes. So what are processes? They're arm-like extensions that extend from the cell body. In the central nervous system, they contain both the neuron cell bodies and the processes, but in the peripheral nervous system, it's chiefly just the processes themselves. So again, we have two terms when we talk about collections of these processes, depending if we're talking about the central nervous system or the peripheral nervous system. If we're talking about them in regards to the central nervous system, we call them tracts. If we call them, if we have these bundles lying outside of our brain or spinal cord, we call them nerves. And we have two types of processes that can make these bundles, and those are dendrites and axons. And they're pretty different in structure and function. So here's a good example of what most neurons look like. There are some exceptions. So you can see here we have our cell body, and then we have one axon, which is this region down here. And we have several dendrites. So all of these are dendrites extending out this way. So we do have one cell body and then one or more processes. In this example, we have one cell body, one axon, and multiple dendrites. The point of attachment, we talk about an axon to the cell body itself. I want to go ahead and point that out now. Where it connects is called the axon helix. We can see down here, in this particular case, our axon is myelinated. So we see these myelin sheaths. We see that these myelin sheaths are accomplished by a Schwann cell. So we automatically know that this neuron is going to be a neuron in the peripheral nervous system. Because remember, if we have myelination on an axon in the central nervous system, that's going to be accomplished by an oligodendrocyte. We still have in our cell body basic organelles that we expect to find in the rest of our cells, so nucleus, nucleolus. We can see some endoplasmic reticulum, several mitochondria, because we need to produce a lot of ATP or energy in order to transmit and move all these ions and transmit signals. Okay, so let's talk a little bit more about some of those processes. So if we're talking about dendrites, uh, they can span, so a neuron can either have 0 dendrites or, up to hundreds of dendrites. So dendrites, if they do have dendrites, they are typically branching severely and extensively. Here's a good example. And off of these dendrites, they can have tiny spikes and further branch, but not extremely long branches. And those would be called dendritic spines down here. Still considered a receptive region or an input region. So that's the same as with our soma or cell body. So let me go back and kind of point that out. If we're talking about our cell body or our dendrites, those are both considered receptive regions because they are going to receive any incoming signals. If we talk about our axon, this is going to be our conducting region because it's going to generate an impulse. So we have 2 receptive regions of a neuron that can receive incoming signals and only 1 conducting region, that's the axon, where it can actually send that signal and transmit that signal. So going back to our dendrites, again, these are our receptive area, one of our receptive areas, and they can convey incoming messages towards the cell body as what we call something graded potentials. These are short distance signals. So graded potentials are very different from action potentials because action potentials are long distance signals. So our receptive areas, like our dendrite and our cell body, they receive graded potentials, and our axon, which is our conducting region, that's going to create action potentials or long distance signals themselves. The dendrites normally contain the same organelles that are found in the soma or the cell body. Okay, so let's talk a little bit more about another process, which is the axon. So each neuron has to have an axon, has to have a cell body, has to have an axon. It can have dendrites, so it could have 0 dendrites or it could have up to 100 dendrites. But the requirement in order for it to be a neuron is basically to have 1 cell body and 1 axon. I pointed out to you on that image before is that where the axon connects with the cell body, that area is called the axon hillock. That will come into play later on when we talk about getting the message from the cell body, those graded potentials, and turning them into an action potential at the axon themselves. So in some neurons, axons are really short or absent, and in others, we're going to forget the absent part because we don't talk about that here. We talk about them in this chapter just in regards to having at least 1 axon. Axon comprises almost the entire length of the neuron, so it's the longest part. Some axons can be over 1 meter long, so about 3 to 4 feet, depending on where they're going, what their effector organ is. And if they're really, really long, we typically call them nerve fibers. Kind of how we called muscle cells that were elongated, we called those muscle fibers. And then, although we typically only have 1 axon, we have areas in which our axon can branch further. So if this is our cell body and this is our axon right here, our axon can branch off of the primary axon. And where it branches and turns into these other axons, we call axon collaterals. So it can branch as many as 10,000 times. And it doesn't matter if we're talking about an axon collateral or the original axon itself, where it lands or where it ends, so all these circles, we call axon terminals. And we talked about this in the skeletal system. The axon terminal was responsible for releasing the neurotransmitter onto the skeletal muscle. So I mentioned earlier that the dendrites in the soma are the receptive or receiving regions, and that our axon is the conducting region of the neuron itself. So how does it actually conduct this? Well, it's able to generate these nerve impulses and transmit them along the axonema. So the axonema is just the plasma membrane of a neuron, and to the axon terminal. At the terminal region, that's where we're going to have our neurotransmitters being able to be released into the extracellular space, which we call a synapse, which we'll talk about in a little bit. And then something that's different from the skeletal muscle is that these neurotransmitters can either excite or inhibit the neurons that they are in contact with. We talked about the skeletal system in chapter 9. Acetylcholine only acted in an excitatory manner towards skeletal muscle. Everywhere else in our body, our neurotransmitters that are released can act on the other neuron or whatever its effector target is, either to excite it or to inhibit its firing. And in this way, it can act to carry on several conversations with different neurons at a time. And the axons rely on the cell bodies or the soma themselves because that's where to produce proteins and membranes, because that's where our machinery is. And if we cut them or if they're damaged, they quickly degrade. So functional characteristics of an axon. Basically, if everything is being produced in our dendrites and cell body, because that's where we have that machinery, we have to have transport in two different directions. When we talk about these directions, we're really talking about down the length of an axon. So you can have anterior grade transport, which is going to be moving things from our cell body towards the axon terminal, or you can have retrograde, which is going to be moving things closer to our axon terminal towards our cell body. So example, again, our mitochondria, skeletal elements, and membrane components and enzymes are all going to be produced in our cell body or our dendrites. So they would use anterior grade transport in order to get those items down to the axon terminal. At the axon terminal, some of the organelles are going to get worn out and degraded, or some of the signaling molecules that we've already used, those will need to be transported back to the cell body. So they would use retrograde axonal transport to get those back there. Other things that take advantage of retrograde transport along the length of an axon are things like viruses and even bacterial toxins. So, a lot of viruses such as polio, rabies, herpes simplex, and tetanus, all of those can travel via retrograde axonal transport and go back to damage neural tissues. So, if we think about where an axon terminal is synapsing, say on our skin, some sort of receptor there, on an internal organ, wherever the synapse is on that organ, it can use where that synapse is to go backwards. And most of these cell bodies, remember, are going to reside in our central nervous system so they can actually go back and eventually hit our brain or our spinal cord. So there's a lot of research undergoing ways to use and take advantage of retrograde transport in order to treat certain genetic diseases so that we can correct things that can get to our brain without us actually having to be invasively going into these areas. Okay, so let's talk a little bit more about the myelin sheath. Remember, the myelin sheath is going to be the result of either a Schwann cell in the peripheral nervous system or an oligodendrocyte in the central nervous system. And basically, all it is a whitish protein lipid substance, and the whole function of it is to act as an insulator. So in the areas where we have the myelin, we do not have the voltage-gated channels or other channels allowing ions to move from the inside of the cell to the extracellular space or vice versa. So if we don't have any movement of ions, which are charge, then that means that in areas where we have this myelin, it's electrically insulating because we can't change the electricity or the charge across the membrane in those areas. So, how does it function, or what's its purpose to be on an axon? Is that if we do have myelin, because it's electrically insulating, we can actually increase the speed of nerve impulse transmission. Myelinated fibers, the ones that are actually myelinated tend to be the ones that are the longest or have the largest diameter, because if we have a really, really, really long axon, we want to increase that speed of nerve impulse transmission so we can get the signal sent quickly. The only way we can do that or increase the speed is to actually myelinate it. Non-myelinated fibers, they wouldn't have the Schwann cells or the oligodendrocytes, and so they're going to conduct impulses more slowly. So typically short distance neurons that don't have to go far can function fine without having myelination. The longer or the farther we have to send the signal, we talked about earlier how some axons could reach up to 3 to 4 feet. The farther or the longer the axon is, the quicker or the faster we need that signal to get there. So we're going to need some myelination in order to increase that rate of transmission. And this should be repetitive. So again, how do we get myelination in our peripheral nervous system? It's going to be our Schwann cells. They are a little bit different from the oligodendrocytes in that a single Schwann cell makes up one myelin sheath. And so how does it do this? Basically, the Schwann cell comes and wraps around the axon several times in what's called like a jelly roll fashion. I'll show you a picture on the next slide. And basically, like I mentioned before, one cell makes one myelin sheath. That's different from oligodendrocytes, where one oligodendrocyte can make several myelin sheaths. Anyways, when the Schwann cell goes and it wraps itself around the axon of a neuron, it's basically going to squeeze and push all of its cytoplasm to the external surface because it's rolling so tightly. So you get what's called an outer collar of paranuclear cytoplasm, which is a bulge on the outside with the bulk of the cytoplasm. It's also going to push a lot of organelles there, so our nucleus would also be in that paranuclear cytoplasm. So here's an example of how a Schwann cell forms the myelin sheath. So it's going to begin wrapping and going to continue to wrap around and then wrap and wrap and wrap and wrap some more. And then it's going to push all of that cytoplasm to the outside so we get that perinuclear cytoplasm. So when we're talking about myelination, again, down the length of an axon, we don't myelinate adjacent spaces. So we do leave these gaps. So if here's a Schwann cell in the peripheral nervous system, we space a little bit and we have another myelin sheath and another one here and another one here. So we have gaps in between. These gaps are what we call nodes of Ranvier, and these are also where the axon collaterals can emerge. So I said the single axon can go off and branch. So if it's going to branch, it's going to have to be at these collateral regions where there's no myelination. I'm not worried about that. So here's the picture we already saw before. We can see our myelin sheaths right here and our nodes of Ranvier, the spaces in between. If we were to have axon collaterals, they would extend out at these nodes of Ranvier right here. And that's very different from how we have the axons in the central nervous system myelinated, because one cell can myelinate multiple axons and form multiple myelin sheaths up to 60. So here's one cell forming several myelin sheaths. So because it's not wrapping itself in a jelly roll fashion, we do not have a perinuclear cytoplasm. I do want to point out, even if it's an oligodendrocyte making the myelin sheath, we still have the gaps in the nodes of Ranvier. So I mentioned what myelin is. It's a lipid protein substance, and it's white. So if we do have myelination in our brain or a spinal cord, we call that area, which you've probably heard of, white matter in the brain. Why? Because it literally looks white because the axons of the neurons are myelinated. In areas where we don't have myelination, we don't have that white lipid protein substance wrapping the axons, and it actually appears gray. So we call that gray matter. Okay, let's talk a little bit about how we classify neurons. If we're talking about structural classification, they're basically talking about how many processes do these neurons have. And remember, your processes are your dendrites and your axons, and typically we require at least one axon. So the image of the neurons that you've seen so far, all of those are what you consider multipolar, and those are the most common type, where we have our cell body, one axon, and then multiple dendrites. We can also have a bipolar neuron, so we just have one dendrite, one axon in our cell body. And then we can also have what's called a unipolar neuron. In this case, we just have the cell body and one axon, and we don't have any dendrites at all. So here's a picture, which is probably better than me drawing it. Again, our classic, most common type of neuron, that's going to be our multipolar neuron, where we have a cell body, a single axon, and multiple dendrites extending off. Here's our bipolar neuron, where we have a centrally located cell body, an axon extending off one end, and then a single dendrite extending off of the other one. We also have our unipolar, so we have a central cell body, but instead of having a dendrite, our axon extends along the whole length right there. Don't worry about this, that's too much information for you for this class. However, down here on this table, you should kind of understand that multipolar neurons are the most abundant in the body. Bipolar are pretty rare. We would find this in our special sensory organs, so something like our sense of smell, so olfactory mucosa, our eyes, and our ears. And when we talk about unipolar, these are mainly found in our peripheral nervous system. And we'll talk about this later on, but not right now. The other way we can classify neurons is functional classification. So how many processes they have, that was structural classification, but what is the function of each one of these? So when we talk about functional classification, there's 3 different types, and they're grouped by the direction in which the nerve impulse travels relative to the central nervous system. So just like we kind of already talked about, we talked about sensory fibers and motor fibers. That's the same thing here. So if we're talking about sensory neurons, those are going to transmit impulses from sensory receptors in our skin or other organs toward our CNS. They're going to approach it because that's our afferent pathway. Almost all of these are unipolar because they're trying to send information in one direction and to one target place, so our central nervous system. The cell bodies are going to be located in ganglia in the peripheral nervous system, so that's just a generic name, again, the ganglia, for a group of cell bodies in the peripheral nervous system. And then our second functional classification is going to be motor. So that's where we're going to carry impulses from our central nervous system back to effectors. So things like muscles or glands, because we're going to probably be synapsing on several different things. Those are going to be multipolar. Most cell bodies are located in the central nervous system, except for some autonomic neurons. The third classification, which we haven't talked about yet, are the interneurons. So we talked about how we use sensory neurons to get information from our senses to our central nervous system. And then we talked about how we interpret that and send it out through motor neurons to an effector organ or gland. We didn't talk about how we actually process that information in our central nervous system. And how we do that are interneurons. So these lie between the motor and the sensory neurons, and they're going to help shuttle the signals within our brain and our spinal cord themselves. So most of these are going to be found entirely within the central nervous system, and 99% of the body's neurons are actually our interneurons and found within our brain or our spinal cord itself. So here's just kind of an example. talking about, and again, I just kind of want you to pick up on the main words or the main functions. So if we're talking about multipolar, most of these are going to be interneurons, so that means they're going to be found within the central nervous system. We talk about unipolar. These are a lot are in their sensory neurons and found in pathways to the CNS for interpretation. We also have some multipolar neurons that are motor neurons. And we talk about bipolar neurons. Those are going to be sensory neurons because those are found in our sense organs that we already talked about, like our smell, so olfactory mucosa, eyesight, and our hearing.